beautiful people. This is JJ, your friend, your mentor. So, I hope that you're all doing good and you're all safe at your places. Today, we are dealing with a topic that is structure of DNA. So, we are in a continuation with the previous lecture where we have learned nucleotide and nucleoside. So, here I have a cute picture of three friends posing together, that is phosphate, the sugar, and the nitrogenous base. So this is known as a nucleotide. That is, when three of them come together, it is known as a nucleotide. Whereas when there's two of them come together, it is known as nucleoside. But which two? Okay, so the two of them are the nitrogenous base and the sugar. Here, the phosphate group is not included. So this is a nucleoside. Okay, as well as we have discussed about the bonds that is formed between the phosphate and the sugar, and also the nitrogenous base and the sugar, that is this and this bond. Look, I'll suggest you that you should watch the previous video because it will help you to understand this particular topic, that is bond formation, in a very better manner. So I'll put it somewhere here. You can just click on that and then you will get a video. Yes. So now it's the recap type where we will discuss about the bonds that is formed between the phosphate and the sugar, and also the nitrogen base and the sugar. So we'll start with the phosphate and the sugar bond. Okay, so here we see that the phosphate is attached to the fifth carbon. Okay, it is attached to the fifth carbon. You can see that here. Yeah, and the phosphate which is attached is the alpha phosphate. Look, the first phosphate is alpha phosphate, then beta, then gamma phosphate. And all this phosphate, that is this triphosphate, is formed from the phosphoric acid. So when three phosphoric acid come together, they form a triphosphate. Okay, so the first phosphate, that is the alpha phosphate, interact or react with the fifth carbon, and that's how H2O is released. Here you can see that H and the second H and O is released. So H2O is released, and this particular bond is known as an ester bond. Okay, you have to remember that this particular bond is known as an ester bond. Yippee! So like that, and then we will move on to the sugar and the nitrogenous base. So nitrogenous bases are basically they belong to two teams, that is team purine and team pyrimidine. So team purine includes adenine and guanine, which base pairs with the other team, that is thymine and cytosine. They do not have any other choice, that is cytosine cannot base pair with adenine, nor can guanine base pair with thymine. Okay, so we'll see the bond. So here is guanine, which means it belongs to the team purine. Okay, so team purine always pairs or makes bonds with the ribose sugar or deoxyribose sugar with the help of the ninth nitrogen. Okay, whereas in the case of pyrimidines, they bond with the help of the first nitrogen. So you have to remember that purines, they form the bond with deoxyribose sugar with the help of the ninth nitrogen. So here we can see that nitrogen has a H group, and of course the first carbon also has a OH group. So when they both come together, a H2O is released, and this bond is known as glycosidic bond. Okay, previously we have learned about ester bond, that is in between the phosphate and the sugar. Here we are talking about a nitrogenous base and a sugar where a glycosidic bond is formed. So this was the case of purine, but in the case of pyrimidines, that is thymine is a pyrimidine. Okay, here N1 nitrogen is bonded to the C1 carbon of the deoxyribose sugar. So in the case of purines, N9 was involved, but in the case of pyrimidines, N1 is involved. So here also N automatically has a H group, and of course the C1 carbon also had a OH group. So I have made a direct bond. This is a glycosidic bond. But you have to understand that H2O is released here also. So that's how the bonding system works, and now we will move on to the main topic, that is DNA formation. So this is our today's topic, and we will just move into that without further ado. Yeah. So a DNA is formed of so many nucleotides. So the criteria for becoming a nucleotide is very simple. It should have a phosphate group, okay, check. It should have a deoxyribose sugar, okay, check. And it should also have a nitrogenous base, okay, check. So it is a nucleotide. So many such nucleotides should come together, and when they come together, they form a DNA. We'll take an example of. So here we have monomers. So many monomers come together and form a polymer, right? Similarly, so many nucleotides come together and form a DNA. Tada! So here this is nucleotide 1, and then comes other nucleotide to attack the first nucleotide and form a bond. Okay, so here is a second nucleotide where the alpha phosphate, so this is the alpha phosphate, this is the beta phosphate, and this is the gamma phosphate, and we know that. So the alpha phosphate's OH group attacked the third carbon's OH group of the nucleotide 1. So this was the nucleotide 1, which has 5 carbons, of course, because it's a pentose sugar, right? So 1, 2, 3, the 3 has a OH group, you have to remember that, and then 4, and then 5, right? This OH is attacked by the alpha phosphate. Okay, so when that attack happens, the first thing that happens is that a pyrophosphate is released. That is, the beta and the gamma phosphate is released, and this is known as a pyrophosphate. So we are just left with a alpha phosphate. You have to forget this part. We're just left with the alpha phosphate where OH is present here, and of course a nucleotide 1 where OH is present here. Okay, so this is what it looks like. So the H, the OH and the OH bonds, and here H2O is released. Okay, and we are just left with the O. That is, the O bond is present between the C of the third carbon of the pentose sugar and the phosphate. Okay, so H2O is released during a condensation reaction. That is when two molecules come together, and when H2O is released, it is known as a condensation reaction. Okay, we have seen a similar kind of reaction somewhere else. Remember? Do you remember that? Yes, here, when a ester bond is formed between a phosphoric acid or a phosphate group and the fifth carbon of the pentose sugar. Here also H2O was released, and it was also between acid and a OH group. Remember? So this is also an example of what? Yes, ester bond. So here two ester bond is present. That is, one is present over here, and the second ester bond is present over here, that we have already learned, right? So this is a phosphodiester bond. Phosphor because a phosphate group is included here, right? And di because two ester bond is present. So 1 and 2. So diester bond. And this reaction is catalyzed by DNA polymerase. I will help you to learn that in the next video, which will probably about DNA replication. Okay, so that's all about the phosphodiester bond. So similarly, another nucleotide, that is nucleotide number 3, comes, and they form a phosphodiester bond, and of course this is what DNA looks like after the end of the process. Okay, so I have slanted this picture to help you understand this in a better manner. So this strand you see, it is formed of the sugar phosphate backbone. Okay, this is formed of the sugar phosphate backbone. Okay, and then we have the nucleotides or the nitrogenous bases bulging out, right? So these nitrogenous bases which are bulging out are present here also. That is A, G, and T. That is A, G, and T. And here one more thing you should know is that there's a 5' end and there's a 3' end. What does that mean? So the 5' end simply means that it is the fifth carbon over here at the top, and it has a phosphate group. So it is usually said that the 5' end has a phosphate group. It's because that the fifth carbon has a phosphate group attached to it. There is nothing above this, right? So it is the 5' end, and beneath at the lowest point of the DNA, here we have a 3' end because a 3 carbon is present over here with the OH group. So the 3' end has a OH group, that is 3 OH end and 5 phosphate end. So when a DNA is formed, they are in anti-parallel form. So if it is from 5 to 3 direction here, then it will be from 5 to 3 direction in the opposite strand. Okay, in an anti-parallel manner. So here the reverse of all this happens. That is, you should imagine it like this. I have reversed the picture. Okay, so here we have a 5 phosphate end and here we have a 3 OH end. Okay, so that's all about the DNA structure, and we have some few questions. That is DNA negative or positive? Well, you have the answer. DNA is negative because of the negative mark or the negative charge on the phosphate group. Okay, so the phosphate group is negatively charged. Okay, it has high tendency to lose its protons, so it is negatively charged, and that's why it is acidic also, because the phosphoric acid is used here. So we have an answer to the second question also, that is whether DNA is acidic or basic in nature. Well, DNA is acidic because of the phosphoric acid that is present in the DNA. So imagine so many phosphoric acid is involved in the formation of nucleotide, right? So if this is a DNA, then we have a phosphate group here, here, and here, and the thing goes on. So we have so many negative charges and we have so many phosphate groups. So it indicates simply that DNA is acidic as well as DNA is negatively charged. And when we are asked about the dimensions of DNA, it might look like this. So the distance between two DNA strands, or the width of the DNA, is 20 angstroms. Okay, and the distance between two base pairs is 3.4 angstroms, and the distance of a helix, that is when it takes a full complete turn, that is 34 angstroms long. So this is 34 angstrom long, and it contains 10 base pairs, whereas here we have just 4 base pairs. This is not true, this is not the case. We should have 10 base pairs in the common DNA, that is bDNA. Now the last question is that what happens to the DNA in our body? So according to central dogma principle, this is a DNA. Okay, this is actually double stranded, but for transcription we have to consider a single strand right now. It complementary base pairs with the RNA. Okay, so here a DNA is base pairing to form a RNA. So A forms with base pairs with U, because in RNA of course uracil is present and not thymine. We have studied that, right? And guanine is as usual base pairs with cytosine, and this is how we get a transcript, that is mRNA, and it further detaches from the DNA. Okay, and the mRNA is now free to undergo translation and form proteins. So we'll see how. Three base pairs come together and they form a codon, and a codon codes for an amino acid. So here ACU base pair came together and form a codon, and this codon specifies or signifies serine, which is an amino acid. Similarly, AUG is responsible for valine. So many such amino acids come together and form a protein, and protein is what we need, right? In our body we need so many proteins, and the most important one is hemoglobin. So there are so many proteins in our body which is involved in our day-to-day activity. So DNA is important because it controls the expression of all our genes which codes for proteins. Yeah, and there are many other important functions. Okay, so it is very amazing that it actually is 2 meters long, that is DNA is 2 meters long and contains 3 billion base pairs. Can you imagine that? How do you think that this big DNA is accommodated in our small cell? So there is a process for that, and we will learn about that right now. So this is our DNA double helix, right? And this is negatively charged. DNA is negatively charged. So DNA wrap around histone. Histones are proteins which are what? They are positively charged. So negatively charged DNA wraps around the histone proteins and form nucleosomes. When DNA and histone proteins come together, they are known as nucleosomes. Okay, then what happens? The nucleosomes coil again each other. Okay, here the yellow thing you see is nothing but histone. You try to remember it as something which looks like a ball. Okay, so the nucleosomes are itself coiled right now, and they form chromatin fibers. So this is how they look. Now they condense further and form a chromatin. Okay, so this is the most condensed form of the DNA, and then further this chromatin is in our chromosome. This is stored in our chromosome. So this big DNA is stored in our chromosome with the help of a lot of condensation, and there are so many proteins involved in this particular process, and hopefully we'll learn about all this in the upcoming lectures. Okay, so thank you all for bearing with me until now, for watching my video. If you like my video, please give it a thumbs up, and of course share and subscribe, because it encourages me. Okay, so thank you for everything. Much love. Bye from JJ.